Okay, so now I am going back to the screen share. I have a few slides for you, and then we will jump into the Qlucore and NDEx presentation mode. Okay, perfect. So what are networks and why networks? So when NDEx tool refers to the networks gene to gene, pathway to pathway, gene to protein, protein to protein, and also gene to small molecules. And this might not even be everything there. It's really a great and large resource. So like right now on the left, here is an example. This is a gene network visualized as genes and pathways. So in blue, I know you cannot see, it's really big one. The resolution is not good enough for you to see if I zoom in here. But blue ones, blue spheres are pathways. And in this case, they focused on signaling pathways. And red are differentially expressed genes. And I think the size is a measure weight importance in this network. And we are looking at 303 altered genes. So I understand those are only differentially expressed genes. And they're connected in a network based on signaling pathways only. So you will see that the conclusion we can draw from this network is that the list of, you can decide like 7 or 8 genes, the largest, represented with largest circles, have the highest degree, highest values, and they are the best candidates for importance for discovery. In this case, it's liver fibrosis pathogenesis, which was studied. So a little more, why networks and pathway analysis are so popular. They are intuitive to biologists, and I speak as I'm a biologist with some training in bioinformatics, computer science, but definitely this is a really important part of discovery for biologists when you're handling your data and your results. It provides more context, excuse me, biological context for your results. And it's a lot more efficient. Of course, we are talking about high throughput data compared with gene by gene search for gene function. This is pretty much not doable anymore with the modern data sets. And display usually is quite intuitive, easy to understand. So what we can do, we can visualize multiple data types on pathway. We were talking about genes, proteins, small molecules, and we can find active pathways, and we can, like this is just one example, identify potential regulators. You can identify, focus on various things here. And one example which I really liked from NDEx publication or one of the presentations about this tool is these networks can reveal meaningful biological information when differential gene expression analysis is not working at all in terms of not giving any picture. And you can read about it. There is an example about autism, like complex disease, right? Multigenic. How we get there, so to do the network analysis. So it can be your own data, or it can be kind of theoretical research when you just have, you want to study some pathways. And then you can go ahead, just get the members of the pathway, and let me show you right now. I prepared a list. That's all you need. In this case, it's IL-2 STAT5 signaling pathway. Simplest text, plain text file, just one gene, human genes in this case. So I would make a copy of this list and take it straight to the NDEx discovery tool. So that's, I don't even have my data, but if I wanted to research this network, this pathway for network data. And the other one, which is more fun, and we all, if we work with data, could do that, is using it for your own data. And your path, sorry, I'm not in the presenter mode now. The path here will be of course, you start with faster files for RNA-seq, then we align faster files. We get BAM files. When you have BAM files, which are aligned reads, right, to genome, we combine them with GTF corresponding, and then we can get expression counts. First, we get row counts, and then they have to go through normalization process or steps of normalization to get normalized counts. Normalized counts will be used for differential gene expression analysis. Then you can run some pathway analysis. GSEA, in this case, I will use GSEA. From GSEA, you will use, or from other tools, you will get a list of pathways altered in your experiment. And then we handpick pathways which we think are of interest, which are changed to the largest degree, and we take them to NDEx. And what this is, the project, just a few words. It's an open source, free for everyone framework where you can store, it's a web, where you can store, share, manipulate, and even publish networks. So you can share privately, and you can share public in a public, just publish. So when you sign up, you get 10 gigabytes of free cloud space. So they say that it's enough to store many of your networks. I don't know exactly how many, but so you can use them as private and also make them available, accessible to everybody. So that's how this resource is growing. And there is a publication. I will share some links in the chat a little later. And just let me see. This tool is integrated with Cytoscape. Probably many of you know and use Cytoscape. So it's really a great, two great resources combined together. I just wanted to give you an idea how much went already into creating this. And this might be a little even outdated information. So what we have there, curated pathways from Wiki Pathways and Signar, published pathway figures from the last 27 years. Also, this really cool machine assembled networks using the Indra system, so artificial intelligence put to work, and then NCI pathway integration database as well. And they state that it's integrated with MCGB, right, which is a very popular, very well curated pathway database. So that's you get access to all of that. Let's go back now to Qlucore. Wait, okay, this is GSEA. So I'm using this data set, which is apoptosis, study of apoptosis in melanoma treatment. So I took it from GRAIN database. This is like a curated, easily accessible in terms of normalization of counts. You get easy access to counts from gene expression database. So it has been curated, formatted, and GRAIN database will allow you just easy with one click, you get the tables for RNA-seq experiments. Now, what we are looking at the biplot sample PCA on the right, 3D and synchronized variable PCA. So those are genes, 33,872 genes. And in my case, I have 9 samples because I limited this data set to control group and MTX 1 micromolar. So I excluded 2 chemo drugs. And also I zoomed in on, this is 0 non-apoptotic control and 10 hours early apoptotic only groups to make it simpler for this, for our case. But so you can easily adjust this all in Qlucore, your inputs. So let's first set up quickly. I will just walk you through the typical workflow, how you would follow here. So if we go to statistical analysis, I will run to group comparison on treatment. comparing MTX versus untreated my control, two-way up and down. And I will start with P 0.05. So you see instant feedback in the software. So all calculations I'm showing are done in real time, and even screen share is slowing it down a little bit. So we're looking at 13,000 results, which so 13,000 genes will pass this stringency on p-value, and then Q, which is Benjamini-Hodgeberg, which this Q corresponds to about 13% of false discoveries. So I can be, if you like, you can be more stringent. And for example, I can, to show you, I will apply Q 0.05, which corresponds to 5% max of false discoveries. And still, I have more than 10,000 genes, and here they are. You can add all these annotations, p-value, q-value right here. So, for example, if you want to look at the top, let's say 100 or something genes, you see everything happens in real time. So top 99. So those are values stringency, which we need to get to the top 99 most differentiating genes, and here they are. You can sort them by lowest Q value, so this is our top candidate. But so that, of course, you can go ahead and plot all kinds of plots here for your results. We don't have time right now to do this, but you will see the heat maps, and let me just quickly at least plot one heat map. And I will configure. So we are looking at all of this 99, only top 99 genes. Let's order them by treatment. We have two treatment groups, samples and genes. I will order by hierarchical clustering. Labels, you can choose what you want to see. I want to plot treatment for now, and gene symbols for genes, for variables, and options for coloring. I will color by treatment, and you can, of course, you can add all other sample annotations which you have at any time. So the top 99 differentiating genes are all upregulated in our chemo MTX, I assume. So just zoom in. Yes, so they all are upregulated in our treatment, and this is an early apoptosis stage. Possibly, most probably, when you look at your results, you know the top players, and you will already see from here which pathways are engaged in this. You will get some hints visually, knowing your genes, but for a pathway analysis, I will take this data set into Geneset Enrichment analysis, which we have integrated right here with Qlucore. And here it is. I will not be running this one in real time. It will take me about, with screen share, about half a minute or so, or just don't want to spend time on this. So I'm showing you what I did in the setup of, I used the complete data set, not just differentiating genes. This is the latest recommendation from the Broad Institute, and I use signal to noise ratio as a metric for comparison. You could also use two-group comparison, but most frequently used recommended by the Broad Institute GSCA team is signal to noise ratio, I feel like. So I apply signal to noise ratio for treatment. Treatment, annotation treatment, and I'm comparing MTX group, which is my chemo treated group. And then the results for gene sets, you need to keep a collection of your own gene sets, and I have here reactome and MCGB pathways. So in this case, I use the discovery kind of bird's eye view hallmark collection in gene symbols, because I have human gene symbols and human gene sets. But hallmark will give me the overview. 50 gene sets are in this collection, and you can see the names on the left. And the results are sorted by normalized enrichment score. So the higher the better, and the most, based on this metric, the most the most enriched pathway here is UV response. You can go, this is the name of the Jeanset from pathway from MCGB. So you can always go back, and there is even the link right here, location, where you can read about this if you don't know. But you will look here in list details, you will see what genes are included in this pathway. And you will see that the size 158 genes are included into hallmark UV response. And in my data set, I have 151 matches. Then you will see normalized enrichment score and normalized enrichment score with P and Q values. And in list details, you can see which genes from in this pathway are driving enrichment, which ones are co-enriched genes. And it's this last column where you will see yes or no. And then what you can do, you will choose, then you have apoptosis and P53. So you will walk through those results normally and pick the ones of most interest for you. I will take this right now, the top 3, and I will export them into Qlucore into the main window, which is right here and in variables. So now you see your data set, this data set, and then 3 gene sets. So what I can do, I can use them as filters. So I just check this box, and we are looking, I need to change my statistical cutoffs. I will clear this. I want to visualize all of the pathway. It doesn't matter significant or not. So I want to see how this UV response genes behave in my data set. And here is the heat map. And this is the variable PCA. We could switch to sample PCA and color it by treatment. This will be more, yes. So we keep this separation. And then you can walk through the other, like now we're looking at apoptosis, how it behaves. So upregulation of apoptotic pathway heavy in treatment, which is not surprising, right? And then P53 pathway. So the same, the part of the cell death. Now this is the point where you will make some decisions about which pathways you want to explore in the networks. So two options. You can export them if you want to keep track as a text file. So you have those this gene set as a text file. You can copy your genes right here. Another easier way is if you right click on the set list right here, you see copy list contents. So if I use this option, then we walk to NDEx discovery tool. This is the main interface where you will enter your gene list. Oops. Yeah, so just paste and you click search. And it will take several seconds. By the way, when you have questions, feel free to put them into Q&A or chat in the WebEx, and I will go over them when we are done. So now we are looking at the results. So first, you have curated pathway. We will see what the genes which we fed into, what pathways they are matching among curated pathways. And often you will have, like right here, you will have quite a long list. This is a very long list. So we need to sort them. So you have several options. Sort by similarity, by p-value of comparing these pathways, comparing pathways to my set, and by overlap. So if you look at similarity, the first hit is direct P53 effectors. So and I'm working, I brought here P53, right? So that's what you see the figure on the right. So there's so many options to work with this, to zoom in, zoom out. You can even drag those bundles, some genes, if you want to make one gene stand out visually more appealing for what you need. And then you have in red are the ones which are present here. And let me show you. So you can highlight query genes, which I just brought into here or not. You see how they offer you different layouts. I feel like default is the best one, but you have options. For example, the next hit here is Wiki Pathways, P53, transcriptional gene network. You see it's the human. Then let's look at what results we get by p-value. It's redoing the analysis. So still, we have direct P53 effectors as our top hit. Then you can explore by overlap as well. Yep. So this still is our first, but then we got different set gene pathway as the second hit. Now let's look at the options for pathway figures. I'm staying in this P53 effectors for now. You will see some figures from publications, and as they state, they keep a collection of 4 figures from like 30 years of publications. So you will have lots of options. Then you can always go and look at all of the genes, and they're highlighted with pink at my query genes. And then there is an Indra Go tool, gene ontology, and this is machine learning, kind of artificial intelligence curated plots, networks, which you can explore. Again, related to, now you will see single transduction by P53 class mediator. So it found the closest network. And you will have access to publications under this interactomes. So you will see a list, like sometimes it's quite short list, sometimes it's longer than this, but you can go and review the existing publications right here. So let me, I will go back to my slides. Just I have two more slides. And so what takeaways at least, what I think, which takeaways I would make from here, that using networks will add a lot more depth, a lot more information for your functional analysis, for your discovery. And these two NDEx powered by Cytoscape, they're great resources for you, and open source as well for networks. And so in terms of the workflow, RNA-seq, and in terms of what you can do in Qlucore, differential gene expression, visualizations can be done in Qlucore. Then we have integrated GSEA, or you can use your other tool of preference for pathways. And then you get pathway lists, which you copy and paste into NDEx Cytoscape. And by the way, if you have Cytoscape on your computer downloaded, you will have an option to move from NDEx directly into Cytoscape. Maybe you are used to Cytoscape interface more, so then you can use that as an option. And I showed you the input, right? So what for, oops, sorry. For the current clients, when you have Qlucore, you can add networks easily to your to your workflow. And for the new users, you can download, I'm not sure why it's moving that slide, sorry for this. You can download Qlucore trial on qlucore.com. And so with RNA-seq data in Qlucore, you can start with either the count of matrix, the matrix of counts, normalized counts, best option, or BAM files with a GTF file bundled together. So that's everything I prepared for you. Let me know if you would like me to reproduce any part of the demo or show anything in Qlucore, GSCA, or NDEx.